Lymphatic vessels have not yet been demonstrated in it, but are represented by the channels in which the bundles of nerves run. These channels are lined by an endothelium. The nerves are numerous and are derived from the ciliary nerves. Around the periphery of the cornea they form an annular plexus, from which fibres enter the substantia propria. They lose their medullary sheaths and ramify throughout its substance in a delicate network, and their terminal filaments form a firm and closer plexus on the surface of the cornea proper, beneath the epithelium. This is termed the subepithelial plexus, and from it fibrils are given off which ramify between the epithelial cells, forming an intraepithelial plexus. The vascular tunic, tunica vasculosa oculi. The vascular tunic of the eye is formed from behind forward by the choroid, the ciliary body, and the iris. The choroid invests the posterior 5/6 of the bulb, and extends as far forward as the ora serrata of the retina. The ciliary body connects the choroid to the circumference of the iris. The iris is a circular diaphragm behind the cornea, and presents near its centre a rounded aperture, the pupil. The choroid, choroidea. The choroid is a thin, highly vascular membrane of a dark brown or chocolate colour, investing the posterior 5/6 of the globe. It is pierced behind by the optic nerve, and in this situation is firmly adherent to the sclera. It is thicker behind than in front. Its outer surface is loosely connected by the lamina suprachoroidea with the sclera. Its inner surface is attached to the pigmented layer of the retina. Structure. The choroid consists mainly of a dense capillary plexus, and of small arteries and veins carrying blood to and returning it from this plexus. On its external surface is a thin membrane, the lamina suprachoroidea, composed of delicate non-vascular lamellae, each lamella consisting of a network of fine elastic fibres along which are branched pigment cells. The spaces between the lamellae are lined by endothelium, and open freely into the perichoroidal lymph space, which in its turn communicates with the periscleral space by the perforations in the sclera through which the vessels and nerves are transmitted. Internal to this lamina is the choroid proper, consisting of two layers, an outer composed of small arteries and veins with pigment cells interspersed between them, and an inner consisting of a capillary plexus. The outer layer, lamina vasculosa, consists in part of the larger branches of the short ciliary arteries which run forward between the veins, before they bend inward to end in the capillaries, but is formed principally of veins, named, from their arrangement, the venae vorticosae. They converge to 4 or 5 equidistant trunks, which pierce the sclera about midway between the sclerocorneal junction and the entrance of the optic nerve. Interspersed between the vessels are dark, star-shaped pigment cells, the processes of which, communicating with those of neighbouring cells, form a delicate network or stroma, which toward the inner surface of the choroid loses its pigmentary character. The inner layer, lamina choriocapillaris, consists of an exceedingly fine capillary plexus, formed by the short ciliary vessels. The network is closer and finer in the posterior than in the anterior part of the choroid. About 1.25 cm behind the cornea its meshes become larger, and are continuous with those of the ciliary processes. These two laminae are connected by a stratum intermedium consisting of fine elastic fibres. On the inner surface of the lamina choriocapillaris is a very thin, structureless, or faintly fibrous membrane, called the lamina basalis. It is closely connected with the stroma of the choroid, and separates it from the pigmentary layer of the retina. One of the functions of the choroid is to provide nutrition for the retina, and to convey vessels and nerves to the ciliary body and iris. Tapetum. This name is applied to the outer and posterior part of the choroid, which in many animals presents an iridescent appearance. The ciliary body, corpus ciliari. The ciliary body comprises the orbiculus ciliaris, the ciliary processes, and the ciliaris muscle. The orbiculus ciliaris is a zone of about 4 mm in width, directly continuous with the anterior part of the choroid. It presents numerous ridges arranged in a radial manner. The ciliary processes, processus ciliaris, are formed by the inward folding of the various layers of the choroid, i.e. the choroid proper and the lamina basalis, and are received between corresponding foldings of the suspensory ligament of the lens. They are arranged in a circle and form a sort of frill behind the iris, around the margin of the lens. They vary from 60 to 80 in number, lie side by side and may be divided into large and small. The former are about 2.5 mm in length, and the latter, consisting of about one third of the entire number, are situated in spaces between them, but without regular arrangement. They are attached by their periphery to 3 or 4 of the ridges of the orbiculus ciliaris, and are continuous with the layers of the choroid. Their opposite extremities are free and rounded, and are directed toward the posterior chamber of the eyeball and circumference of the lens. In front they are continuous with the periphery of the iris. Their posterior surfaces are connected with the suspensory ligament of the lens. Structure. The ciliary processes are similar in structure to the choroid, but the vessels are larger and have chiefly a longitudinal direction. Their posterior surfaces are covered by a bilaminar layer of black pigment cells, which is continued forward from the retina, and is named the pars ciliaris retinae. In the stroma of the ciliary processes there are also stellate pigment cells, but these are not so numerous as in the choroid itself. According to Henderson, the aqueous humour is a secretion formed by the active intervention of the epithelial cells lining the apices of the ciliary processes. End of Section 50.